I'm the head of facilities management for the Betsi Cadwaladr University Health Board up in North Wales. A very complex name and a very complex organisation. And I'm joined today by my colleague Andrew Bullock, who's one of our hotel services managers, and Andy's actually been supporting the trials in regards to the use of hypochlorous acid within the organisation. So the presentation today, we're not professional presenters, yeah, we're operational managers, so please bear with us. They did tell us that there'd only be about 2 people here, but yeah, it's great to see everybody. Thank you very much. So yeah, we want to talk about our case study and where we've been looking over the last couple of years in regards to alternatives to the use of HPV. So, Betsi Cadwaladr University Health Board, shortened to BCUHB. What are we? We're a health board up in North Wales. Lovely place, Snowdonia, Rill. We've got all the kind of attractions that people want to come to. And actually the health board provide a health service to around 700,000 people across North Wales and the many visitors that come to the area as well. We cover about 6 counties across the top of North Wales. So it's quite a big geographical area. It's equivalent to, for our district general hospitals, equivalent to a hospital in Liverpool merging with a hospital in Manchester, merging with a hospital in Leeds. So anywhere else that wouldn't happen, but it's happened in North Wales. Okay, so we've got 3 district general hospitals, we've got community hospitals, we've got health centres, GP practices, mental health units, dental surgeries, community health centres. We've got a prison, one of the biggest prisons in the UK. We've got around 134 wards and about 2,400 beds. And we're an integrated health board. In regards to our facilities, or our facilities teams, which I actually head up, they're all in-house services, so we don't have any outsourced services in Wales. So we've got a really large team that support that across our domestic portering and catering services. And that equates to somewhere in the region of about 1,800 staff. Okay, so we've been looking at innovation for quite some time. I suppose our journey started back in 2012, where we first trialled HPV, hydrogen peroxide, and we've actually kind of grown the amount of equipment that we've got over that time, where we've trialled different systems and we've brought different companies in actually to support our HPV program. So we started that in 2012. We started to look at UV back in 2013, and we presently have 9 HPV machines, and we've got about 18 UV single towers that are in operation across the health board. We've also trialled the robots, so we had them on trial for some time. We did some development work with robotics in regards to UV as well. So we've got a really, really good understanding of a lot of the HPV systems and UV systems that are out there on the market. And we've also got a really good agreed methodology for the environmental cleanliness and use of these actual pieces of equipment. And one of the big elements for us is actually making sure that we're using the right equipment for the right purpose. And we've actually introduced a system what we call What Clean Do We Mean, which actually highlights when we need to use HPV, when we need to use UV, and also when we need to use conventional cleaning to actually support our decontamination processes. Okay, so let's move on to the interesting bit, the case study around the use of hypochlorous acid. Why Metis Health? You know, why Hydra? Well, Metis Health came to us initially to actually ask whether we would do some research work with them to actually look at the product that they've got and actually look at is hydroperoxide, you know, the kingpin of what we use, or is there alternatives that we can use? And that's where we started to look at the use of hydroperoxide. And I think the first thing that came to mind was, one, it's really safe, where HPV is quite dirty in a lot of ways. I call it kind of a dirty process because you have to have somebody sitting outside the door whilst you're actually undergoing that process. You have to make sure that the rooms are sealed. You can't use it in certain areas. It does escape through the ceiling time to time and actually come down in the next room. I'm sure that people have actually had issues around the use of HPV and the safety relating to it. So the first thing that we thought was actually this is really good and it's really safe. When we looked at the efficacy data, we could see that, you know, this is too good to be true, to be quite honest, because it seems to do the same as HPV and it seems to do the same as UV. Yeah, but actually it's electrified salt. You know, so we started to get really, really interested around the kind of research work that had been done with it. And one of the big questions that we had was where we've looked at hypochlorous acid in the past. Yeah, it's actually been an advert on Amazon for a kettle that'll produce it, and it's got kind of a shelf life of about 6 hours. Where all of a sudden we've got a stable product which we can actually use, which has got a shelf life of, you know, 12 months. So these are all starting to make us think about let's trial this and let's kind of do a little bit of work around it. I think the other element why we picked with Metis Health was, you know, they were really assured in regards to the time saving versus the use of HPV. You know, and my colleague will get onto it in a minute. I don't want to ruin his thunder, but, you know, we did a single room with ensuite facilities, you know, and not including the pre-clean, because you still need to physically clean with any of these processes. But the actual decontamination process time was about 17 minutes. So then it started to make me think, this is HPV, an alternative to HPV, but this is starting to become an alternative to UV. There's also different methods for actually dispersing the product as well. So we were looking at misting. You know, it comes as a spray. You can get it as a wipe. And there's also the options to actually look at self-generation of hypochlorous acid on site, which, you know, for me, having a one-stop solution to all your cleaning products is really quite innovative. And the other reason why was actually the only company that has got the rights to sell this in healthcare in the UK, and I've got all the regulatory documents to actually support that. So it was actually a bit of a, you're the only one, so we'll come to you. So that's the reason why we went to Metis. Okay, can I hand over to Andy? Is that okay?
Yeah. Thank you, sir. So, as Paul said, my name's Andy Bullock. I'm Hotel Service Manager for Betsi Cadwaladr University Health Board. I've been with the organisation for 30 years. I like to think it's 25, but it is actually 30 years. And I have to say the last 2 years that I've worked there have been the most challenging years out of the 30 years. And as part of that process, when we first went into COVID, we were struggling with staffing, we were struggling with time elements, we were struggling with everything, with the demands that were put on the service. And I finished my shift at 5 o'clock and I went home and I came back at 9 o'clock to support the deep clean team by sitting outside the ward bay for 3 hours whilst the HPV process cleared the air. The fogging had happened, it was just literally waiting for that area to become safe. So whilst I sat there, I've seen a lot of things on the wards going into COVID, and it was fantastic because I had 3 hours to actually think, what am I doing here? What am I doing stood outside a bay with poisonous gases on one side, me on the other side, and a piece of wood that my staff had taped up, hoping that nobody's going to go in there, no gas is going to escape, and everybody's going to be safe. And when I thought about the risk assessments and everything else that went with that, I started to panic. And like I say, it's been a challenging 2 years. Before the 2 years, I had hair down to my shoulders. and I come out of it this side. But to come out of COVID and to be part of this, when Paul asked us to trial this, that was really a key point for me, because I'd had that experience of what our staff on the wards have to go through, and they just crack on with it. So we started the initial trial. We had 2 rooms on our paediatric wards, the children's wards in Bangor, Ysbyty Gwynedd. Both rooms were cleaned. We didn't ask the team anything more than to do a full clean on the areas, the 2 rooms. We swabbed the rooms afterwards with ATP and with micro swabs, and then we misted the rooms, 1 room with 1 product, one with, sorry, not with a product, one with 1 machine, one with another delivery method and another machine. We then re-swabbed. We had the ATP protein results straight away, immediately, and we sent away to a lab for the micro swabs to be returned. So we picked on, as I said, high touch areas, and if we'd all been listening to Tom McCulloch earlier, everything on there will now make sense to you earlier with the counts, and literally we did each area as a high touch. You can see on the left there's the pre-misting ATP swab test, and on the other side, and it's fair to say everything came down with the ATP misting. There's some high areas that we saw on the left there that, you know, we'd done the clean, and for us, we'll get to it later, it just goes to show that however due diligent your staff are, areas will get missed. And I think you know, we've done the micro swab test there again, and with every single one of them there has seen a reduction in the log count and the biological log count that was presented afterwards. So I won't go through everything. I think it's quite clear there that and for me, it's yes, it's a fantastic piece of kit. Yes, it does it. Paul touched on it earlier, the time. It takes us 6 hours to do a ward bay with the HPV, and we can do exactly the same area with this piece of equipment in just over half an hour. And for us, with the turnaround of the wards and the expectation that we need to get bums back in beds after an infection, it was just one of the major concerns that we had, and one of the challenges that we had throughout COVID. So, like I say, the micro swabs reflect more or less the same as the ATP swabs. The same for the results for room 2 with the ATP swabs. And like I say, exactly the same solution, just a different method of delivering that into the room. The results for micro swabs, I think with this one, there was a couple of areas that obviously from the pre-mist, they have not been cleaned properly. You know, and for me, the bed rail and the spine holder, you know, you know, and it happens. It will happen with our staff, and with everything that they've gone through, the expectations of them. It goes to show we're not 100% perfect, but I've got to say as well, to be fair, the standards of hygiene of our cleaning from the results that we had pre-misting and post-cleaning were showing that the standards of hygiene are high. Some places were missed, as I've said, through the routine cleaning and disinfection. The misting solvic through the mist array reduced counts to less than 10 in all areas in room 1, and the same process in room 2 reduced it to 10, less than 10 in 66% of the area in room 2. Mystic solving confers additional benefit above traditional spray and wipe disinfectant, because again, you're still relying on the actual physical clean and making sure that every area is actually cleaned physically. The misting mitigates areas that have been missed during the routine cleaning and the disinfection. And for us now, we are looking at a programme, once we're up and running with this system, of every clean we do, we should be able to put this machine in, and it's just for us a backup of 30 minutes to make sure that we've, you know, what we've done physically, we're just backing it up and making sure that everything is done. So once we'd done the test, the trial at Bangor, we moved over to our colleagues in Glenclwyd Hospital and Clarke Hospital, and we did again in Ysbyty Gwynedd. We did 2 general wards, a communal bathroom, 2 high dependency units at Bangor, 2 rooms there, and we did 52 swab sites. We pre-cleaned, post-clean and post-mist were where we took the swabs. In Clarke Hospital, which is a community hospital, we did it on Cadwaladr ward, and again asked the domestic team to clean. The reason we've got the second one there, the room had been cleaned previously, was just for time, because we, throughout the whole of the trial, trying to find a ward or an empty room in a hospital during COVID was more or less impossible. So the side room was swabbed, it was cleaned with usual detergent and Actichlor Plus. It was re-swabbed, misted with the solution, and 2 minutes to mist it, 5 minutes dwell time, and it's not been changed on there, but there is another 5 minutes there for demist as well. And exactly the same, the wet room was swabbed, misted, and we're looking at, again, 15 minutes. Again, the trials from Clarke came up with the same results, a considerable reduction in log count, and I think for me that was what we've been waiting for, is the confirmation, isn't it, Paul, that, you know, I'm not a microbiologist, I'm not a chemist, I don't know anything regarding, you know, the efficacy, and that is what we need to know. And for me, the data is there, and it's quite clear. There was a post-clean in the middle there with the underbed buttons, which we presume we contaminated it during the process, and again the misting brought it back down anyway to a safe level of less than 10. Just in a graph form, you see that on the left is the start of the process, and everyone apart from the last one is a reduction down to nothing apart from as well the second. In Glenclwyd Hospital we did on Ward 2, and the swabs were taken from Bay 6, which had again previously been cleaned previously for timing. The room was swabbed, misted, again 20 minutes maximum, and the final swabs were taken. I say again with the 4 minutes, the 5 minutes, and we cannot get away from the pre-clean. What we can get away from is the preparations that you have to make through the HPV of taping up the room, securing the room and making it airtight. And with this particular product, it's safe. It doesn't matter so much if there is an escape of it. Again, we did find that once any fog did come from the room, there was a mad panic from the rest of the ward, we're on fire. So lesson learnt, we still do tape up the doors, but we don't have to be as due diligent with doing that, because it is 100% safe. Again, Glenclwyd, the reduction there, and it shows for me the back of the bed remote control, the reduction there, it was shockingly dirty. And I think, again, shows with our staff, remote control, you clean the buttons, but your hands are actually around the back of it whilst using it. So again, although the figures were significantly high, there was a significant reduction in the log. And again, just in a graph form. In Ysbyty Gwynedd, we did 2 HDU rooms, and again, we swabbed. On this occasion, we swabbed. We did the clean with the domestics, we re-swabbed, and we misted the room again for the same time. And again, a 15-minute, 20-minute process. With the Ysbyty Gwynedd one, there was 3 rooms together. What we found was, whilst the team had cleaned the first room, normally they would then have to stand and wait, put the HPV processor on and stand outside the room, and that is dead time. What we were able to do with this is, because it's completely safe on the other side of the wall and through the door, they were able to leave that room, just put a sign there to say no entry, and then they were able to start cleaning the next room, and we actually got 4 rooms done in less time than it would have taken to do 1 room with the HPV. And again, and it's the data is there, and for me that is all that mattered, was getting the data from the laboratory back to say that you know, it brings it all down to a safe level. Again, so post-misting on only 4 of the 52 swabs showed any counts of any significance. With those 4, there was an average 3 log reduction, again, which is a significant reduction on the pre-misting counts that we carried out. In high dependency, 100% of the swabs were less than 100 per swab post-mist. Post-intervention, better at every stage. And we did find, probably with the HDU, because of the area, there's a subconscious factor from our staff that this has to be cleaner than any other part of the hospital. That's our perception, but that's what we deciphered from the results. So as a summary, standard of cleaning is very high across all 3 hospitals. The cleaning removed most of the service bioburden, particularly when the cleaning intervention was performed intermediately prior to swabbing. Rooms that had been cleaned prior to our arrival had slightly higher initial microbial burdens, possibly due to recontamination of the area in the lag time between them being cleaned and us arriving to swab. In HDU, normal cleaning resulted in 86% of the samples returning less than 100 CFU. And after misting, this rose to 100% of samples again. The current cleaning regime, detergent plus Acticlaw, is thorough. It's effective, however, and like I've said already, the misting of the solvent confers additional microbial reductions on a surface where things may have been missed or recontaminated. And for me, more than anything, it's the time scale of having that in place and being able to put that in place to ensure that if anything has been missed, we've caught it again at a later stage, but not taken 6 hours to do it. Thank you.
I thought you were going to carry on then, Andy. Leave me sit down. What Andy didn't mention was 2 years ago he was also 6'2" tall. So just to pick up on a little bit of what Andy's talked around there, I think one of the interesting bits around this was when the team come to actually do the swabbing and then went away and then presented the results to us. They said, actually, your cleaning regime in your hospital is really good. And one of the questions that we do have is, well, you know, how good? Well, they were saying that, you know, we were probably around the log 2, log 3 area in regards to, you know, the hospital in general. So it makes you question, doesn't it? When you talk about HPV, you talk about UV, and they're talking about being able to achieve cleans around log 6, log 7. Actually, do we need to actually put that type of kind of chemical into our buildings to actually ensure that we've got them fully decontaminated. And I think that's a question that we need to reflect on as part of this discussion. So in regards to Solvic, as I said, you know, one of the stumbling blocks for hypochloric acid was around its stability. The product that we're using is stable. It's got a 12-month shelf life on it, so we've got plenty of time to actually use it. But the product itself, you know, they're saying that it's 300 times more powerful than bleach. You know, that's one really big statement, isn't it? But actually, because of the way that hypochlorous acid is made up, it's really gentle. You know, so we find that in some countries, it's used for acne care, it's used for iron or oral health care. So actually, if it's used in those areas, yeah, it's probably pretty safe to use around our staff and patients. So yeah, as I say, you know, it's a natural product. Our body uses similar technology to actually fight bacteria. So it's not a new innovation. This has been around for billions of years, hasn't it? We've been using it as a human being for a long time. What we've actually managed to do now is put it into a system that, one, is stable and that we can use. We can actually use as a misting process, you know, and I think that's the only difference why we haven't been using it for a long time. So, how does it work? It's fast. It's simple. It's highly cost effective compared with the use of HPV. It's pH neutral. As I say, it's very effective. You know, I think Andy has presented the stats to actually prove that. And there's no clinical resistance to it. So that's the system we've got in front there. It's fully under, all the bits to it are very much kind of tried and tested and proven through all the trials that we've done. And the product is very much proven through where it's used in the food industry and other industries outside of healthcare. I know that the Hydra system is being worked on, and now trying to make it, you know, a system, an all-in-one system that looks like a BMW. So, you know, it will be very sleek and it will have a really new livery, and that's what Husain has been telling me during the conference today. Really easy to use. You put it in a room, you turn it on, yeah, you let it do its job. You go back in the room, you put the room back together, and it's as easy as that. It's literally around pressing buttons. And one of the things I do like about it is that when Andy was talking about sitting outside a door for 3 hours before, what we have to have is a lot of trained staff to use HPV and also to use UV. So if you haven't got trained staff, yeah, you actually can't use the process. And we've had times over the last 2 years where we've had a bed out of action for, you know, 4 or 5 days because, yeah, we couldn't actually get an HPV team into there to actually undertake the process. And this has been really compounded in a lot of our community hospitals, yeah, where we've had patients and literally we've had to then put equipment in the back of a van with a deep clean team, send them out there, do the process, yeah, and then obviously we can then reoccupy that room. With this process, it's so easy, it's so safe that we can actually let anybody use it. So, you know, our aspirations is to make sure that we can, you know, get as many people trained, our domestic assistants trained, housekeepers trained in the organisation. So actually it becomes the norm. It becomes part of the daily cleaning process. So in conclusion, Betsy's been working around the trial and the use of hypochloric acid now for around 18 months, 2 years. You know, and I think we've had really good success and we've got really good evidence around it. We want to do a lot more trialling with the use of hypochlorous acid against C. diff and see what the kind of outcomes are against particular elements. The I can't see that one. Okay, I should say this. The Health Board has initially purchased 8 systems. Yeah, and as I say, our aspiration is to actually not only use these in our acute care, but actually to make sure that we've got them in our community hospitals so that they're free and ready for use, so that we don't come back to the issues where we've got bed blocking for 4, 5, 6 days because we can't get an HPV system out there to actually undertake that clean. You know, by actually kind of making sure we've got enough systems in place, yeah, that should help support us in that way. And I suppose we started the question by asking, you know, is Hydra Solvic an alternative to HPV? In my view, yes, very much it is. But I think what we've also learned that we weren't actually looking at the 18 UV systems that we've got because we thought, well, you know, they're quick, they're easily used. But actually, yeah, when we look at it, you know, hypochlorous acid is an alternative to HPV and UVC. Okay, thank you very much. Thank you.